I will be introducing Professor Luigi Preziosi, who is going to give the Trami lecture. Afterwards, we will have the pleasure to give him the Trami prize. And before letting Professor Preziosi start, I would like to tell you something about him. Something which I took from the Laudato, which is available on the website.
Not working, I guess.
Of MEM. So you can have the full text there. I'm going to take some parts of that text plus something which I will add. So Luigi Preziosi is full professor of mathematical physics at our Department of Mathematical Sciences, Giuseppe Luigi Lagrange, at the Politecnico di Torino, in the northwest of Italy. And he is the leader of our group, which is called Group of Models and Methods in Mathematics. And Luigi received two PhDs, one in mechanics with minor in mathematics, and one in mathematics afterwards. And he became professor in Italy a few years afterwards, in Torino, then in Calabria, then again in Torino. And I think I can say that Torino is the place where he remained for the longer time and where he's going to remain for a long time. With several students. And of course, a man with his career has achieved many things to be proud of, but I think personally that one of the best recognitions that a professor has, the best results ever, are his students. And so I can say that among the qualities of Luigi, maybe the best one is the capability of raising scientists at several levels, depending on their aptitude. So he's had many students at the level of bachelor, master of science, doctorate, or postdoc. And the majority of these students were able to have a very good career, either in the industry or in the university, in the academy. And this is, of course, possible if you dedicate your life to raising students, and if you have many different interests. And in fact, the scientific interests of Luigi range from industrial mathematics, in the real sense of the word, to biology, biomathematics, and in both fields he gave very big contributions. I'm not going to give the report the full list because it is available on the website. I would just like to recall that in the field of industrial mathematics, Luigi gave a huge contribution, which also got in a patent about sand dynamics, so the dynamics of sand in the certain zones, which is a very huge problem for railways built in those regions. And in the field of biomechanics and more generally mechanobiology, he is recognized for the contributions that he gave primarily in tumor dynamics, tumor growth, angiogenesis, and for the collaborations that he established all through the years with medical doctors, with well-known institutions like the hospital that in Piedmont we have in Candiolo, where a lot of research on cancer is conducted. And everybody knows how bad cancer is and how important the contributions in this direction are. And Luigi gave, or has been giving, because he continues to give contributions in this field, and they are very important. So I would like to say that the scientific production and the quality of this research brought him to receive an important recognition, which is the membership to the Accademia dei Lincei. I don't need to recall that the Accademia dei Lincei is very, very important. In Italy is very, very important, not only in Italy. He is the member of several scientific boards, and maybe now a personal thing. I believe, as I began to say when I started my speech, that what qualifies a professor mainly is the capability of being a mentor. And a mentor, depending at which level you enter in his or her life, is what the professor teaches you or is able to give scientifically. And I got acquainted with Luigi when I was already grown up. So I was not his student formally, because I entered this group as an assistant professor at the end of 2011. And but we collaborated a lot, and he spoke few words with me that gave very, very precise directions to my own research. And I think this is very important. Sometimes you don't need to talk to somebody for years. Sometimes it is sufficient that you give just few words, which are fundamental, and your scientific life changes. And this is what happened to me. So the majority of my research descends from a discussion occurred on a train while we were going to a conference. And I'm very thankful for that. And so I'm glad to introduce you now, Luigi, and to listen what he is going to say to us. Luigi, thank you very much.
Okay, thank you. Thank you, Alfio, for this wonderful introduction. Not really deserved at all, but I mean, it's always a pleasure. What I would like to present to you today is something that attracted my attention during COVID, which has to do with this phenomenon. The experiment has to do with seeding cells on a substrate and then pulling on it. Okay, and what you see is that according to how you pull, you have that these cells reorient themselves, either in shape, and then we will see also internally. Most of the literature, they say that the cell orient perpendicularly to the direction of the principal, say the direction where you pull, or at a certain angle. Okay, and in fact you have this sort of distribution function, of course symmetrically, because you don't have an up and down, and so this is the point. Why this is of interest, and why I started doing that? The interest starting in the 80s, when people wanted to understand why in the artery the cells tend to align according to very, very specific directions. This is a graph, for instance, by Zapfel, in which you have the distribution of the cell, say, axis over the angle, or perpendicularly. I mean, longitudinally along the direction of the vessel, of the aorta, for instance, of the blood vessel, or if you want, perpendicularly to the circumferential direction. And the interest for that is also to build tissues, say stent-like, if you want, replacement of the arteries, but also on the heart, because the heart is the same. I mean, the heart is a very complex structure in which, say, the fibers, myofibroblasts, orient along very specific direction. Also in depth, they change. And so if you want to build a patch of myofibroblasts to put on the heart to have a to cure for infection, for instance, then you have to build it properly. You have to control it, right? So this is the main idea. So the amazing thing is that they performed till the 80s a lot of experiments. Actually, in the 80s, they started. The blow up of this kind of experiments, say, is about, say, 2000 and 2010, I would say. And they use different kind of cells, okay? And they so that for all these cells here, they see the same thing. I mean, and these are very different cells. You have muscle cell or myocytes, as we discussed before, but also osteoblasts, endothelial cells, or whatever. There is only one example, which is, however, a negative example, which is, however, instructive, which is the macrophages. The cells of the immune system don't do it. And the reason for that is that the difference between these two very large class of cells have different roles in the environment. Namely, the macrophages, or in general, cells of the immune systems, have to crawl the environment, go very fast. Their motion is very, it's more or less, it's called amoeboid. It's blobby-like. I mean, instead, these other cells, these other cells here, they attach to the substratum, to the extracellular matrix, they pull a lot, and so on. So they have a strong, say, inner structure, a strong cytoskeleton. And in fact, this is more or less a picture of the cytoskeleton, and these fibers of actin that form the cytoskeleton and also of other filaments, microtubules and so on, they form a network that is connected by a number of proteins, say filamin, or say there are this, from the mother, say filament, you have daughter filaments at a certain angle, and there is a molecule here, Rp23, that governs, in some sense, this angle here, which is close to 70 degrees in rest condition. And so you understand, while filamin more or less take the filament and say the rest angle is about 90 degrees. So you understand that when you pull the cell and you try to close this angle here, there is an elastic response inside the cell. And also, the important thing, actually, the most visible part in the cell is that when you do these experiments, they form stress fiber. Stress fibers are just a bunch of filaments of actin that are held together by a number of proteins, fascin, actinin, and so on. Okay? So these are the structure, and so you respond to that. This is one thing, but the other amazing thing is that the phenomenon is very robust to a lot of things, with respect to a lot of things. Say, cell type, of course, substratum stiffness, in the sense that as long as the substratum is stiff enough, you get it, and the angle of orientation is the same, or amplitude of how much you stretch, as long as this is big enough, and stretching frequency. Stretching frequency, by the way, means that, for instance, if you take a specimen and you just stretch it, and you keep your substratum deformed, you don't see it. You have to periodically stretch, like the heart, like the artery, okay? At a certain frequency, okay? So what I would like to cover here, to give you, is an explanation of this phenomenon and also of this robustness with respect to all these kind of things. In some cases, the answer is quite good. In other, probably there is more work to be done. Okay? So let's look at the problem from the continuum point of view. And a specimen, and assume that you have a deformation that is very simple. The deformation gradient is diagonal. Okay? And the important thing here in all the discussion that we will make is this biaxiality ratio. Okay? So is minus epsilon yy over epsilon xx. And what is found, these are the set of experiments that is more complete, are those by Livne, who actually controlled in a biaxial test this biaxiality ratio, this R here, and found that if you plot the cosine square of theta of the angle over this strange number, 1 over r plus 1, you get more or less a straight line, okay, for different value of s, or of r. And to understand what's happening here, this value, which means this r equals 0, means that we are clamping the sides, the y sides in some sense, the upper and lower sides, and we are just stretching along the x direction. In this case, you have cosine square equals 0, which means theta equals pi over 2. Okay? Then, and so the cell orients perpendicularly as described in a lot of papers. Then you have the other extreme in which you have that r is 1, and you have the angle that is 45 degrees, and this one, which corresponds to, say, an incompressible material. And you have a lot, in fact, you have a lot of experiment here. Okay, and the angle here is more or less 60 degrees. Okay, so how can you do that? And why is this independent of everything, I would say, from the mechanical point of view? So let's start from a very general, say, constitutive equation. You have to take into account that your energy, in some sense, depends on several invariants, anisotropic invariants, that all have a role in defining the response of the cell with respect to the deformation. I don't want to enter in detail because probably most of you know that a lot. But the thing that I want to show is the following, that if you look at this invariant in the plane, because the experiment is two-dimensional, actually they are just function of cosine square. And this gives you already a hint of why the y-axis is cosine square equals something. Okay? The only thing that does not depend on cosine square is this one, is 8, but because of the symmetry, you have that the energy has to be even in I8, and so you go back to something that for any energy that you find, any energy that depends on all this function, this is just a function of cosine square. That's it. So if you want to find the equilibrium orientation of this energy, you just compute the derivative. What you find, trivially, is that either you have the horizontal orientation, okay, horizontal I mean along the main stretching direction, or the perpendicular one, when the cosine goes to 0, and or possibly you can have that this becomes 0 because u prime, the derivative of u at cosine square is equal to 0. And this gives this thing here. Actually, in order to do that, I have to specify a bit more the energy that I'm using. So let's take, for instance, a very general quadratic elastic energy. I can use also a Fang energy, it's the same. And what I find is that u prime goes to 0 when cosine square is equal to 1/2 plus something. Okay, and this something depends on the mechanical characteristics of the material. Okay, so everything is, all the mechanics of the cell and of the substratum, by the way, are contained in this coefficient here. Okay, so K66, K44 takes into account of the response of the cell with respect to the pulling along the main axis of the cell. I don't want to say polarization, but say orientation. There is no motion here. Eh? They are just static, and they just orient, and they form this inner structure with the actin fibers that are aligned along certain direction. And for instance, K66 governs and describes the response with respect to deformation in the perpendicular direction, and K88 has to do with the shear, the response to shear. So this is the mechanics, and this lambda here, this quantity here, is related to what you are doing to the cells and to the substratum. So how much you are stretching along the x direction, how much is compressing along the y direction. And this, you can call it lambda. And in the linear regime, this capital lambda is 1 over 1 plus r. And so you find very trivially that it's natural that for every energy, or if you want, not every, but a large class of energies, you have a linear relation between cosine square and 1 over 1 plus r. Okay? And this explains the robustness of this thing. Okay, let's go. The only thing that can change is this angle here, alpha. Okay? This angle, this slope here, alpha. But before I go to that, let me observe that here, the way you are stretching, you are deforming the substratum is described by this r here. And this r is the ratio of the stretching along y and x. Okay? And so it depends on the ratio, not on how much you are stretching in x. So it means that you have the same angle if you are stretching 10% along x and you, say, deform it by 1%, the strain y is 1%, or you double it. You double both. Okay? You get the same thing. And this, in some sense, gives you an idea of why the angle, the final angle, is independent of whether you are stretching 1%, 2%, 5%, and so on. The second thing is that focusing on this part, this is the part that is tested experimentally. It corresponds to, say, stretching in one direction and the specimen is shrinking in the other. Okay, what you have is that you have the theta equals 0, the horizontal, say, the parallel orientation is unstable. The perpendicular one must be stable in this range, and you have a lot of oblique orientation according to r. R or lambda is the same. Okay? And these are bifurcation points in terms of stability analysis, which turn out to be supercritical. Okay, coming now to the parameter, because you see, alpha depends on a lot of things. So you may ask, what is the minimum ingredient that I have to put into the model to get this result? If you forget about everything and you assume that just, say, the response of the cell to the formation along its main axis is important, then this number is equal to 1. 1 means that you have the yellow line, which is not the one that is seen by Leibniz. I will come back to that in any case. It's not the one seen by Leibniz. Leibniz sees something that is, say, around 0.8, which means that there must be something else. This something else might be, there is a relation between the coefficients that must be satisfied. And one thing could be K88, could be important. That is the response to shear, because K88, of course, lowers the denominator, and you can also evaluate how big that is, 0.2. 0.2 of K44. Okay? This is one way. And another way is to add, say, coupling between, say, for instance, I4 and I1, the trace. And you have that, in this case, this coupling coefficient is 0.06. I mean, at the moment, I don't have any Q to tell you which is the same. Certainly, it cannot be K66, because if you include K66, you have that this number becomes larger than 1, which is the black line. Now, the question is the following. In most of the experiments, what you see, actually what you read, is that cells align perpendicularly. Okay? So this is an experiment by Wang, more or less 2000, on endothelial cells. The description is just, say, in words, but he has some graphs. Okay, he does 2 experiments when R is 0.34, which is more or less here, and they find an oblique orientation with a certain distribution function around, say, 70 degrees, and R equals 0, which means they clamp the size. R equals 0 is here, okay? If, according to Leibniz experiments, they should be perpendicular, but you know, if you look closely, both at this figure here and at the histograms, you see that it's not really probably 90 degrees. It's something less. So it might be, it's not excluded from the experiments that are done, that endothelial cells have a different R, a different alpha. Okay? So they might have a non-negligible case in response to the perpendicular direction. In fact, endothelial cells are more round. I mean, the distribution of stress fiber is not so aligned as in, say, fibroblasts that are used by Leibniz. In the fibroblast, the cell is very elongated, more or less triangular, and all the fibers are in one direction. Instead, endothelial cells, they are more round, and so the distribution of fibers over, say, all the direction, there are fibers in all directions. Of course, the most of the fibers are in the here, 85 direction, but you cannot exclude there are other fibers in the other one, which are the one that are responsible to the response to the perpendicular direction. Okay.
Why are you singling out that red line? Because
the red line is the line by Leibniz. Okay. The line that is fitting the experiments by Leibniz that has as all the points are lined here. Okay, so this is the only line that you find in the literature, and I'm assuming that this picture here corresponds to this point. I know that this is R equals 0 means that I'm here at 1, and if it is not 90 degrees, means the cosine square is something. So it's not down here, it's somewhere up here. So it might be the blue one, the black one. Okay, just this. Okay, this has to do with the equilibrium configurations. How do you go to this equilibrium configuration? There are some other data. These are still some evolution from different initial angles of this orientation. And if you, this is when you change take a single cell that initially has, I don't know, 30 degrees and will go toward the equilibrium value. Of course, it can go to more or less 60 degrees or to minus 60 degrees, depends on the initial condition, it's just the same. And this instead is what happens when you change the biaxiality ratio. The equilibrium is different, but more or less the timing to go there is the same. It is of the order of, say, minutes, actually more or less 1 hour, okay? Half an hour. Okay, so in order to do that, what you write is just an equation that tells you that theta tends to minimize the energy, a gradient flow in some sense. And if you do the simulation, what you find is that it's very similar to what you get in the experiment. Okay? Nothing else to do. I mean, I just added to the U, the general U that I was using with that alpha, if you want, with that parameter, just an evolution in theta. And the only thing that is calibrated, if you want, here is eta that takes into account of the timing to go there. Okay, so you fit that with one experiment, with one line, and then you use that for the others, and you are in a correct shape. And these three lines here, these three curves, correspond to these middle curves over there. So also the timing is good. Okay, let's go now to the other aspect. The fact that this is very robust unless, I mean, you have to take into account that amplitude and frequency have to be large enough. Strain amplitude. Well, one thing that you can observe is the following, is that if you, in the experiment, you increase the amplitude, you don't change the equilibrium, but you change the timing to go there, okay? So the characteristic time to go toward the equilibrium decreases when you increase the amplitude. When it's below 1% here, you don't see anything, or you see very little. And there are a couple of explanations for that. The first one is very rough. The first one is related to the fact that if you look at the right-hand side of this ODE, you find out that there is a factor epsilon xx squared, okay, in front of it, which means that the timing to go toward equilibrium scale with the square of the strain that you are imposing. And in fact, if you take some numbers, these are the timing of your orientation of the cell body by this experiment in just a couple of years ago. And let's start from this, 15% strain, takes 10 minutes. 10% is 22 minutes. So this is what? 2/3 square is 4/9. And so that's why you find that here, this timing is doubling, is 9/4 of time 10.5. Okay, so in some sense, this is okay. I mean, this is theoretically according to this estimate. And this is what you get if you, say, superpose this timing on the graph. This is the good part. This is the bad part. I mean, in the sense that if you go on, and for instance, you you halve the strain, then according to this reasoning, you should increase 4 times the timing to go there, and this is not true. I mean, this goes to infinity in some sense, which, by the way, from the point of view of a mathematician, is much better to have something that goes to, I don't know, something, and you say, okay, it's disappearing. So there is something else to do here that is not really clear. Why here is not, is faster than expected. So this is not satisfactory. Stretch frequency, the same thing. These are just the same guys. You see how long does it take to get to the equilibrium. You have a plateau at very large frequencies. By the way, this is 1 second. 1 second. Second to the -1, which is what is the period of the heart beating. So that's why you use 1 second as the period of your impulse oscillation. So above, say, this frequency, for very high frequency, you don't find any difference. Below it, you have that this increases. By the way, this is a log-log plot. So this increases a lot. And again, you don't see anything when your frequency is too low. Okay. Actually, this kind of model that I was presenting here cannot explain that, because of course we are in the range of elastic materials, and the elastic response is quick in some sense, it's immediate. Okay? Here, what you need in order to understand why imposing a periodic stretch of a certain period, you have a response. If you have another response, if you impose another period, cannot be included here. You need to include another intrinsic time. And the reason, biological reason for that is that actually there are some phenomena inside the cells that, of course, include an intrinsic response time of the cell. And this involve, say, the cytosol that can be viscoelastic. And so you have, say, a relaxation time, a characteristic relaxation time related to that. But also the remodeling of the actin cytoskeleton inside the cell. And also the turnover of the adhesion bonds between the cell and the substratum. So the picture is the following. The general one is that you are stimulating the cell at with certain deformation with a characteristic time. If these deformations are fast, then the cell does not have enough time to remodel and reorganize the actin cytoskeleton, these bones. Instead, if they are low, they have enough time to remodel internally and to, say, reorganize the addition to the substratum. Okay? So this is the picture. And by the way, if you look at the timing of this, characteristic timing of this phenomenon, they are of the order of minutes. Okay? More or less 1 minute is the integrin turnover at the membrane. Okay? And this is what is more or less the critical time of the threshold where you see or don't see anything happening to the cell. Okay, so in order to do that, we have to use a more complex model, of course, some elasticity. So this is the viscoelastic model. This is the stress, which is a response which takes into account of some of the memory of the strain tensor. Oh, sorry about that. Okay. This is the relative strain tensor at time t and a past time tau. This is the elasticity tensor that depends on the orientation, on the main orientation. And you weight that with the relaxation kernel that we use exponential. We take it exponentially. Okay? As is known, when you use a viscoelastic model with an exponential time, you can rewrite this integral equation as a set of differential equation. One that gives you the evolution of theta in response of, say, the stress and the strain, if you want the energy. And then this is the constitutive equation, which is of Maxwell type. Okay? So this is the relaxation time, the characteristic time that the intrinsic time of the cell response. So if you go to the limit of very fast, in the high frequency limit, very fast, say, deformations, you can actually prove that the response is elastic. This is classical. That is, you can forget about this term here, and the main term is the stress is a response in phase with the periodic stress. You put of evolution for the angle of the orientation, and you find what? You find the equation that I was describing before. So this means that this tells you why when you have a fast deformation, then the behavior of the cell is described very well by an elastic model. Okay? Instead, if you go to the other limit, a low frequency limit, then the response is viscous. Okay? So the stress is more or less related to the time derivative of the strain. So you have another type of response, and you don't see much. These are the same plots that I showed before in the high frequency regime and for this viscoelastic model. The equilibrium values are the same. And this instead are for the lower frequency regime. And what you see is that, I mean, it takes a lot. Actually, you can compute what happens in, as a function of your imposed, say, frequency of deformation. And you see that if this is big enough, you go toward the equilibrium. After some threshold, you don't go anywhere. Okay? And there is a behavior like a phase transition. So for low frequency, the velocity of orientation is much longer. I mean, not the time of orientation is much longer than the time of experiment. And probably noise and random effect in the cell. I will not have time to show the kinetic model that we were working as well. Randomness hides all the reorientation phenomena. Okay? Becomes more important. Instead, for high frequency, you have this transition that occur more or less here. Okay, at one, say at one with a period of 1 second. Okay, So this is the moment in which you start seeing something, and this is the model, time over compared to the experiment, which is much better than the other, by the way, much better than the strain response. I mean, we are quite satisfied with that. Okay, explains a bit of why this timing of response depends on the frequency. Last part, substratum elasticity. Because what is seen, as I told you, is that the substratum has to be stiff enough. Most of the experiments are done on almost, I don't want to say rigid, otherwise you don't stretch it, but you know, plastic material. Okay? Some of experiments are done instead of gel, collagen gels and so on, which are closer to biological materials, of course. They are difficult. And what you see is that in some cases, when the substratum becomes too soft, either you don't see or you see something different from what I discussed till now. I mean, the cell orients along the stretching directions, okay, or perpendicularly, but not with an oblique angle. And this is quite strange. Well, in order to do that, I mean, we found it very difficult to use a continuous model, so we schematized the cell as, say, struts and spring and dashpot, if you want. Actually, not dashpot, springs and torsional springs. So the model is this one. You have two elements, two elastic elements that at rest have an angle of 90 degrees, and there is a torsional spring here. So the cell elastically responds stretching in this direction, in this direction. They attach to one point, one addition point, and so when the substratum is stretched, you have to imagine that when the substratum is stretched, if there is no cell, it would go to just the deformation gradient times x, but the cell will just respond to that, so will pull on it, and so you have an elastic spring here between xa and fxa. You do that, you compute the internal energy. Internal energy, I mean the energy that is related not to the substratum, but the energy felt by the cell. I mean, how much the actin filaments are pulled, actin filament. The elements, this element here is pulled, how much this one is pulled, how much this angle that was 90 degrees, the filament, the one that is related, for instance, to filament, is closing up. Okay? You do that, and you find what is the trend toward the minimum. And this is the equation that you get. What you can see that here is that there is epsilon square, as before. I mean, this is just very similar to the equation that I discussed before. In the limit of Ks that goes to infinity, say, stiff substratum, you just get the same equation that I got before. Now you have epsilon square, you have cosine square, everything becomes function of cosine square, plus times sine cosine, the same as before, but there is Ks that is coming up. And Ks is affecting both here, in front of everything, and these coefficients here. Okay, in a way that is not mathematically complicated, it's just boring to handle. However, the first thing to notice is that there is a Ks here. So when Ks goes to 0 again, this process is very slow. Like when you have epsilon that goes to 0. I mean, becomes the characteristic time scales with Ks square. I mean, scaling with Ks square means that if I lower by 1 order of magnitude Ks, which is typical, I mean, I go to, I don't know, 1 megapascal to 100 kilopascal, which is typical, or you go even below that. I mean, the time, characteristic time becomes 100. 100 means that if it takes 1 hour, it will take 100 hours. And this is not tested in the experiments. And again, in these 5 days, 4 days, randomness plays a relevant role and will hide everything. Just because of that. But the second thing that I was mentioning is that the equilibrium change in this case. The equilibrium change in the sense that you don't see any longer the equilibrium angle, you see only the parallel and the perpendicular angle. You can do it, you can look at the equilibrium of this function. This depends on a bunch of parameters. The most important one are this k perpendicular and k theta. But I will not focus on that because there is Ks. I'm interested in how the cell respond when I can change the substratum stiffness. What I get, okay? No, sorry. This is when I don't have Ks. I have k perpendicular and k theta. And what I want to show you here in this graph is that you jump from a condition in which, in this region above the thick black line, you have a supercritical bifurcation, and in particular, the fact that the equilibrium, the oblique equilibrium is stable, okay? If you jump over this, the black line, this stable equilibrium becomes unstable. And the only stable equilibria are the parallel and the perpendicular one, okay? So this is the picture. From this, you see that it is possible to have situations in which you don't have a stable oblique equilibrium. And I want to translate that in terms of Ks, in terms of the substratum stiffness. And in fact, you find out that this Ks, so here you are in the region of stiff substratum, if you want. And in this, you are region 1. Region 1 means supercritical, means that the equilibrium is stable. Instead here, below this blue surface, in particular soft substrata, you have that the equilibrium is subcritical. So if you try to lower the stiffness and you go from here to here, you have a transition, if you want catastrophic, from an equilibrium that is perpendicular, for instance, and then becomes, for instance, parallel. Okay? And this is, in fact, is what happens. Focus on, for instance, of this value of Kb. Kb is the other parameter, which is important. You have that if I start here, I have that the cell will go toward, say, pi 4. Then they go to pi 6 here, then to 0, and then either to 0 or to pi over 2. Okay? And this is also depicted here. In this small region, you have only parallel, say, orientations. And in this region where Ks is very small, you have either parallel or perpendicular, which is what is seen in the experiment. Okay? Effect of cell elongation. This is the last, more or less the last slide. We gave a look at that. Why? Because, as I told you at the beginning, fibroblasts are very long, for instance. The aspect ratio can become even, say, 5 or 10 times, 5 or 10. The ratio, how long they are with respect to how wide they are. Instead, the nuclear cell or epithelial cell are more round. And what is shown here is that if you change this aspect ratio, so small lambda means that the cell is longer. Lambda equal 1 means that the cell is round. This is what I was discussing before. What you see is that the region 1, the region with an oblique equilibrium, is much wider. Okay? And a region where you have, say, this parallel or perpendicular orientation in soft substratum becomes much smaller. So the picture is that cells, fibroblasts, for instance, are more prone to achieve an oblique equilibrium, and endothelial cells instead might have this change of behavior for soft substratum. So I think that this is the last one. And of course, I have to thank all the people that were, my co-workers that were involved in this project that took a couple of years. And I'm here ready for your questions. Thank you.